All right, AMP1 students, in this video we are going to talk about hydrocephalus, which we didn't get to talk about in the last video. And then we also didn't talk about the primary functions of the cerebrospinal fluid, so we're going to go back and talk about that too. So in our textbook, first we'll talk about hydrocephalus. And so this is a disorder or disease that is caused by excessive cerebrospinal fluid, like too much cerebrospinal fluid in those spaces. And remember, if we get too much cerebrospinal fluid in those spaces, it can push against the bone, but the bone's not going to move. So that fluid, it will put pressure on the brain and could cause brain inflammation and trauma. And so this can happen for a lot of different reasons, but it seems to be associated with some sort of obstruction of the flow of cerebrospinal fluid in those spaces that it's located in, or sometimes there may be a problem with those arachnoid villi, so they're not able to drain that cerebrospinal fluid like they're supposed to. So if we have this case in young, like newborns, whose they still have their fontanelles, then if they have those fontanelles, when they get that increased or excessive cerebrospinal fluid, it can cause movement of those skull bones because they haven't fused together yet. They still have those soft spots called the fontanelles. And so what you can get is an enlargement of their head because of that excessive fluid that's in their spaces around their brain. That can still, though, cause some pressure on the brain too, which could cause neurological damage. There is acquired. This is kind of congenital hydrocephalus, where it occurs at birth or right after birth. And then we also have the acquired hydrocephalus, which can occur anytime, after childhood. And there's a lot of different causes for that. The treatment's kind of still the same. You try to provide a way to either relieve the obstruction or find another way to drain that fluid if, say, it's a problem with the arachnoid villi. So a lot of times they'll put little shunts in, which are tiny tubes, and then the fluid has a place to go. Usually they drain those shunts or little tubes into other body cavities, like we'll talk about the abdominal cavity is one area that they can drain that fluid into. So that way it doesn't put pressure on the brain anymore, and so we can avert any sort of pressure on the brain causing any sort of neurological damage there. So. All right, so that is the disorder that we wanted to talk about when it comes to cerebrospinal fluid. And then up here, which I kind of forgot in the last video, was to talk about like the three main functions of the cerebrospinal fluid. So I already talked about how it's kind of a clear, kind of a light yellow to colorless liquid that circulates in the ventricles and subarachnoid space and the central canal of the spinal cord. But its functions, we already kind of talked about protection, so that's definitely there to provide protection. It's just like a little liquid cushion that can protect our nervous tissue from any sort of sudden movements. And so that's a big function. Another one that you don't probably ever think about, I didn't really think about this, was it provides buoyancy. So our brain is, sitting in the bones, surrounded by the bones of our skull, and gravity alone causes our brain to, want to kind of fall down onto the floor of that skull cavity or cranial cavity, which we don't want it to do because that means our brainstem and those structures on the inferior part of the brain would be squished and pressure would be put on them. So the fluid, if you think about it, we have fluid in kind of surrounding the entire brain on the outside and on the inside. So the brain's kind of sitting in this little fluid environment, kind of like we sit in the uterus and the amniotic fluid kind of floating around. The brain's kind of doing that in our cranium too. And so that gives the brain buoyancy, so it allows it to kind of float and kind of bob up and down in our cranial cavity. And that prevents kind of, opposes the gravity force, so it doesn't get crushed under its own weight by falling to the bottom of the cranial cavity. So buoyancy is another function of the cerebrospinal fluid. And then the last function that we'll talk about is environmental stability. So what that means is it does have a role in, even though the blood is the primary fluid in the body that will circulate nutrients and get them to certain cells of the body and take away waste products, the cerebrospinal fluid can transport some nutrients like the blood does, and even some chemical messengers like hormones and things like that. And it can remove some waste products too. So it kind of has like similar functions to the blood. So it can kind of maintain the environment around the brain tissue. So it kind of gets rid of waste products so they don't stimulate those excitable neurons, and it can bring some nutrients to those neurons that need it for their normal functioning too. So it does that as well. So those are the three main functions of our cerebrospinal fluid that we want to know about. All right, so in the next objectives, we're going to talk about the blood-brain barrier. So we kind of mentioned this when we talked about astrocytes at the beginning of chapter 12. We talked about glial cells, and one of those glial cells was astrocytes, and one of their main functions was to help with forming the blood-brain barrier. So we're going to go into a little bit more detail what that actually means. All right, so our blood-brain barrier. Okay, so the first thing that we want to talk about is what is the purpose of the blood-brain barrier. So realize that obviously we need blood to reach the brain because our neurons and glial cells, they need various components of the blood, nutrients, in order to function properly, and they also need to get rid of their waste products too. But remember from chapter 12, I pulled this up so you recall, we talked about the characteristics of neurons, and one of the characteristics is they're excitable. So they respond to a stimulus. So if there's any change, whether it's a chemical, some sort of stretch or pressure, that can cause them to have a change in their membrane potential and initiate a grade of potential or action potential, essentially. So knowing that, when it comes to the blood may contain various substances that may not be in the right amount, maybe be in excess, or something that could excite our neurons when we don't want them to be excited. Also, our blood carries waste products out of the body. Usually it'll eventually make it to the kidneys, and we'll excrete waste products that way. But while it's circulating, even to the brain, some of those waste products can be in amounts that could, again, excite when we don't want neurons to be excited. And there's certain drugs, too, that could excite neurons when we wouldn't want them to either. So the body needs something to control a little bit more, have a little bit more control on what components of the blood can actually make it to the neurons and into that nervous tissue. So what was created was this blood-brain barrier to kind of keep things out of the nervous tissue that could excite those neurons and cause them to send signals when we don't want them to, and could damage them too. So that's what the blood-brain barrier is doing, is protecting the brain from contents in the blood that could cause them to be damaged or to excite them when we don't want them to be excited. So the way that the blood-brain barrier works is there's two components, and we've already talked about one of them. That was the astrocytes. So the astrocytes were one of the glial cells that are just located in the central nervous system, and they were there to wrap their little processes, foot processes, around a capillary and help form the blood-brain barrier. So they are one component of our blood-brain barrier, the astrocytes, and specifically their little foot processes. And then the other part of the blood-brain barrier are these specialized capillaries. So these are like the tiniest little blood vessels that lie next to our nervous tissue. And these little capillaries are special because their wall is a little bit more resistant to things passing through it. So normally we want certain things to pass through the wall of the capillary, like nutrients, so they can go to the neurons, like glucose would be an example. Neurons need glucose for energy, make energy. But we want some of that to go through, but we want a lot of stuff, most stuff, to stay in the blood and away from those neurons. So the capillary wall is very impermeable to a lot of things that other capillaries in other parts of the body are not. And so they have these nice little tight junctions between the cells that make up the wall of the capillaries, so there's no, very little things can pass through those little slits in there. They're a little bit thicker, their wall is, and again, it's all to try to keep a lot of stuff that's in the blood to stay in the blood and not leave and surround the neurons or be in close proximity to the neurons. So they have a picture, which hopefully it'll help a little bit. So this is showing a capillary, specialized capillary. It goes all the way here, but you can't. It's kind of being covered by these little foot processes of the astrocytes here. So this is you can see a little red blood cell in there. So this is the wall of the capillary. So there's these these different cells that are making up the wall. So there's nice little tight junctions between them, so very few things can slide through there. And then even if some things get through there, then they'd have to contend with the little foot processes of the astrocytes. You can see how they surround the entire capillary here, and only provide these like little slits where things can make it through. So they're kind of making that wall that's pretty impermeable to a lot of components in the blood. So only lets certain things out. And this is a picture. They just kind of cut the capillary, did like a longitudinal section of it, so you can see the wall from this view with your little cells that make up the wall. Little tight junctions between them would be here. Thicker wall as well. I don't really have anything to compare it to, but this is thicker than most capillary walls. And then the little foot processes from the astrocytes. So that's providing that barrier to protect our neurons from components of the blood that could harm them or cause them to be excitable, initiate a action potential when we don't want them to. All right. So the rest of our and then we talk about here how it's not completely, even though it's a nice barrier and it does protect our neurons, it's not absolute. It's not like certain things can't get through there. Maybe we don't want to. So it's not an absolute barrier. So there's some things, and they put a list of a couple of them: nicotine, alcohol, some anesthetics. Those are lipid soluble, so they can more easily cross that plasma membrane of these little cells that are in the wall of the capillary and get into the area, interstitial fluid, that's the fluid around the neurons, get in there. And so they could cause some effects on the brain, on the neurons. So nicotine, alcohol, definitely we know they do. And then some of the anesthetics. So the only place that you won't find the blood-brain barrier, or at least it's greatly reduced in its ability to protect the neurons, is in places where we need contact with the blood.